So yesterday we covered, just real briefly, about mutations, right? Actually, we talked about germline versus somatic first, something about mutations, how to name them, and then we got a bit into DNA extraction, how to get the DNA out. So today I'm going to take a step back further away from DNA and go a little bit more global. And just to quickly review again the types of mutations that can occur. And this time I'm going to ask you to organize it in a way that kind of starts from big to small. So what's sort of the biggest changes you can have in terms of mutations or genetic changes? Okay, so translocations will be pretty big, yep. Anything bigger than that?
Chromosome loss or?
Yeah, so like aneuploidy, right? And even beyond that, do you know what that would be? That's polyploidy. So if you have 69 chromosomes instead of 46, that's sort of a polyploidy kind of thing. If you have single individual chromosomes that are gain or loss, and I think this also applies to maybe a P-arm or a Q-arm as well, that's aneuploidy. You obviously have translocations that can be rather large, right? So what would be under this?
Inversions.
What's that?
Inversions.
Yeah, inversions can be here, yep. Keep drilling down. Remember the next one? So you have your CNVs, right? So remember, greater than 1,000 bases is what we define. These can be a megabase, they can be 2 megabases, they can be a few 100,000 bases. But CNVs technically are also your duplications and deletions, so large ones, so large duplications and deletions. What's the next one? A smaller version of this would be your indels, yep. So get familiar with this terminology, because with next-gen sequencing, a lot of people would say indels, and that's what they mean. Or they're smaller sort of changes. Hundreds of bases, 1,000 bases is where these would be. What's next? The very last one. Point mutations, right? So these are your point mutations. And the other word for that is, you remember?
SNVs.
So SNVs, right? So single nucleotide polymorphisms, or SNPs. Okay, so we're going to start today on trying to pick up some of these things right here, the larger things. We'll be more concerned about this maybe in the third or fourth lecture. But today, let's think about very large changes and how you can detect them. So there is a technique that was developed probably, the very archaic version of it was probably developed in the 1800s, late 1800s, and then it got refined and got quite a bit better for maybe clinical application, perhaps in the 1950s. It's a very old technique. You know what I'm trying to...
Karyotyping?
Yes, so karyotyping. So karyotyping involves essentially a field called cytogenetics, where you can visualize chromosomes at the cellular level. And what does that entail? What would you need for that in order to visualize chromosomes? Chromosomes are chromosomes are quite small, right, relative to the cell themselves. And so you would probably need a microscope. A light microscope is what people have been equipped with, 1950s or so, in order to visualize whole chromosomes and what they're doing. But there is a number of sort of features that are required for you to actually perform karyotyping. First of all, do you recall back to biochem, molecular biology, about different phases of the cell cycle? So there are two that I would like to review. One is interphase, right? And the other one is metaphase. I won't force you to say the other ones, but these are the two main ones that you should be concerned about for this lecture. Interphase, you remember what that state is?
When the cell is not undergoing reproduction, pretty much.
replication and division, right? So they're sort of in their resting state. So in the resting state, it needs to produce certain proteins, etc. So the DNA tends to be a little bit loose, right? Relaxed, loose, and in a state that allows for gene expression and therefore protein production. So this is interphase. If you were to take any tissue in your body, your liver, your muscles, a lung cancer, for example, most of your cells will be in the resting state, in the interphase state. Those that are actively dividing or replicating will be undergoing mitosis. And part of mitosis is there are 4 main cycles, and one of them is metaphase, right? Interphase versus metaphase. So metaphase is one of the 4 steps in the mitosis cycle. And do you recall in metaphase what happens to the chromosomes? So I talk about DNA being loose here in the interphase step. This is probably the one step in a subsection of the cell cycle, a subsection of the mitosis process where the chromosomes are the most condensed. So you can imagine here's the cell here, the nucleus. So in interphase, you can imagine that things are lightly unwound. You still have your nucleosomes probably, right? We talked about histone, slightly about histone code and modification yesterday, how things can be loose or open, and how that affects gene expression and the ability to have, say, transcription factors accessing some of these sites for producing some messenger RNA for some particular protein that the cell needs. In metaphase, by this point, the nucleus has probably broken down. So I'm going to break down the nucleus, sorry. And then you still have your cell membrane here, but the cells are at a state where they're maximally sort of organized. They've replicated, but they're maximally organized and getting ready to undergo cell division. So you can imagine your 46 chromosomes are all kind of lined up here and ready to be divided. So this is when the DNA now, instead of being all unwound and loose, this stuff here gets even more tightly wound and then condensed into a structure called your metaphase chromosomes. And that's when they're ready to divide into the daughter cells. So this is important because if you were to take a microscope and trying to visualize this by microscopy, you're probably going to be way out of luck to even see it, for one, without any staining, or maybe even with giemsa staining, which is the traditional method of staining chromosomes. But you'd be hard-pressed to know what this region is versus what that region is versus what that region is. So in order for you to do karyotyping, you have to have your cells in metaphase, because that's when now you can stain, and the chromosomes will look a certain way depending on what they are and how they band, and that's when you can actually do karyotyping. So in order for you to get to this step, it's almost inherent that what I just told you is you took any tissue out of the body, right? Most of them will not be in metaphase. Most of them will be in interphase. So what does that mean in terms of a prerequisite in order for you to perform karyotyping? Right. So you need to coax them to divide, right? You need to force them to leave interphase, go into mitosis, and divide. And this thing can happen really quickly, very, very fast. As it goes, through the 4 cycles of mitosis, it can go through this thing really, really quickly. So what do you have to do? You kind of have to freeze them in time, right? In order for you to do karyotyping, because it might organize like this, and in a split second, it's already gone into cell division. So you have to force them to grow in culture, to divide, to undergo mitosis, and then you have to kind of lock it in place in metaphase. So this is achieved by using colchicine. You guys remember where this might be from or what it does? It's a drug, it's a microtubule inhibitor, and it's commonly used to treat gout. And the way it does it is just kind of locks microtubules in place. So here, there will be microtubules kind of, they're structures. The centrioles, right, and the microtubules kind of organizing at the metaphase level. It basically locks all this in place and just freezes the cell in time so that it's stuck in metaphase. An alternative is colcemid, which is essentially basically the same thing, a microtubule inhibitor that will force it to be frozen in space. So you coax it to divide. You arrest it in metaphase with these reagents. And the next thing is basically you need to kind of blow up the cells a bit. And this is by using some hypotonic solution. So you may remember from chemistry about gradients and how water goes from lower concentration to higher, right? In terms of salt content. And so if something is, the cell will be hypertonic relative to the hypotonic buffer. And what happens is the water will swell up the cells, blow them up, and kind of make the chromosomes spread out a little bit better. So they're not as tight, they're a little bit more loose. And then in this state, the cells are almost ready to burst, essentially. And the next step that you would do in terms of performing the karyotyping process is you will need to fix it. So you'll fix it in some methanol acetic acid. I think it's like 3:1 or something is the ratio. Methanol acetic acid buffer. And this basically preserves the cell. First of all, it kills the cell, but it also preserves the DNA and all of that so that it's nicely fixed for you. So the next thing is you would actually take these cells. So essentially now you have a suspension. It could be in a test tube. So here's the hypotonic swelled up cells with the metaphases inside. Fix in the methanol acetic acid solution. And you literally are going to do a spread by dropping the cells. So you literally use gravity, drop the cells. So let's say this is my glass slide here. You would drop it at about 10 inches, 12 inches or so, drop by drop. And when it hits the glass slide, it basically bursts the cells open and gives you a spread of the metaphase chromosomes. So ideally, this should be at a certain cell count or number, not too concentrated, because on the glass slide, you will want them to spread out so that perhaps one cell is represented here, another is represented there, another there, another there, another there. And the chromosomes, depending on how well your hypotonic solution worked in terms of swelling up the cells and helping loosen up the chromosomes a bit, you would want the metaphase chromosomes to spread out very nicely and individually like this. You do not want, for example, crowded chromosomes because in this situation you won't be able to distinguish what chromosome is what. So this whole process is actually it's sort of a science, but a large portion of it is very much an art because what affects this method here, this step here, is the humidity of the day, the ambient temperature. Folks would actually blow on the glass slides while they're doing this to help increase the humidity, just so they can get a very nicely distributed metaphase chromosome spread like that. So it's almost like an art where folks with 10, 15 years of experience would probably do it blindly, but whereas someone like us coming in, performing for the first time, would probably fail miserably to get a nice metaphase spread. So I guess what I started out to list here is you need a certain number of criteria to actually even perform karyotyping, right? So you need to have the cells undergo cell division, which means you need what type of tissue?
Not fixed. Fresh.
Non-fixed. So it has to be live tissue, right? So from the body, it would be a tumor specimen. Let's say, for example, a tumor specimen that you just acquired from the OR. You quickly have to put in RPMI medium or some happy buffer so that they can stay alive. You quickly send it to the lab, and literally within hours, no more than half a day or something, you have to start processing the sample and getting them to go into culture. If not, the tissue sits around for too long, the cells will be dead, and you won't be able to grow the cells up. But even if you have live tissues, some tissues are more amenable to tissue culture than others. So for example, some tissues, you can culture them in vitro on the glass dish or plastic dish, but certain other tissues cannot do that. So it depends on your disease and the cells that you're working with. Some will be able to be grown in culture and some will not be able to. So culture is another condition. Culturable is another sort of thing that you have to do. And we'll talk about the other things about karyotyping in just a bit, but these are sort of the two key things. Is live tissue and it has to be cultured. Or grow in vitro are the sort of two main things. Okay? So what can you do with a karyotype? I need to probably speed through this because we need to get to fish after this. So with a karyotype, you essentially have your sets of chromosomes all laid out like this. Sister chromatids, all beautifully laid out. And depending if it's acrocentric or not, the chromosomes have sort of different sort of sizes. And what happens is that when the chromosomes are condensed down, and let's say you do your dropping already, right? And you have the spread. The chromosomes are condensed into their metaphase sort of form here. The next step that you do is you do a giemsa stain, which is essentially the same stain that we use up in hematology to do peripheral blood smears, for example. It'll stain certain things that are acidic, a certain color, and something that's very basic like DNA will probably stain darkly, like with a purple sort of color. So it's a giemsa stain. So a lot of times the most common technique is called G-banding because it's using a giemsa stain for this. So banding means that when these things are condensed, certain parts of this chromosome will stain a certain way depending on the content of the DNA. So those that are very tightly wound, I think, tend to stain a little bit lighter, and something that's very open probably will stain a little bit darker. And it depends on if it's heterochromatin or euchromatin. But this gives a certain pattern. And so if you go and look at chromosomes, they kind of have almost like a barcoded kind of like look, like this. And to a seasoned cytogeneticist, they can look at this based on the size and based on the banding pattern, they can tell you, yep, that's chromosome number 2. And in fact, they can tell you that's chromosome number 2 and that's P21.5 or something like that. That's how good they are. And that's how bad we are, because we, for the first time, look at this, we have no idea what it looks like and what it means. So it takes a very well-trained eye, very well-trained technician, very well-trained cytogeneticist to be able to pick out what is missing and what is not. They may be able to look at this and tell you that perhaps this is the normal pattern, but if something is missing right here, they can tell you that probably there's a piece of chromosome missing here from chromosome number 2. So based on this information, there's a number of information you can gather out by looking at a metaphase spread. So today I mentioned earlier on about copy number detection, right? Which is basically what this is, what that is, and what this is right here. So polyploidy, aneuploidy, and CNVs are all copy number related. So let's do polyploidy. It's very easy for you to look at an individual cell that has been spread out via their chromosomal spread and metaphase spread, and be able to tell, just count how many chromosomes are there, right? So if you see 69, and that's something that's completely abnormal, or if you see 46, that's something that's normal, or if you see 23 for some reason, you can easily detect polyploidy. That's the easy stuff. Aneuploidy comes into play because this is probably the first method of detecting trisomy 21. Is to use karyotyping, and today it's still used as a way to confirm trisomy 21. What is that? That means you have an extra copy of chromosome 23. So if you look through and you count, oh, I have an extra 47th chromosome, and it just so happens the banding pattern, and it's acrocentric, and it looks like chromosome 21. So that's an easy sort of diagnosis for folks to make. So you can basically count the number of chromosomes that you have, and then, like I mentioned before, you can probably detect a subsection of this chromosome being missing here by looking at the karyotype. Just by knowing what the pattern is, with an experienced eye, you can be able to tell exactly what piece is missing. But that exactness is kind of relative. And so if you don't remember anything about karyotyping here in terms of process, I want you to remember one number, which is the resolution of this assay. Any guesses as to how small can I detect a change that that is, say, deleted or duplicated.
5 megabases.
Yeah. So 5 megabases is typically what I kind of quote. Sometimes, yes, you can probably get down to 3 megabases if you have very loose DNA, very good banding patterns. So, like I mentioned, this is an art, right? In terms of the humidity and the temperature, how well the chromosomes are relaxed or condensed. So if it's a very relaxed chromosome, you can probably get multiple, lots and lots of bands. But that's not on a typical day. So I would say on a good day, maybe 3 megabases. On a bad, poor day, no less than 5 megabases. What is a megabase? Yeah, so this is 10 to the 6. So 5 million bases. What's the next one up? Do you remember? Or gigabase, right? This is a billion. Any idea how big our genome is? Say both copies, mom and dad's.
3 to 10 to the 9. 3
Yep,
times 10
so
to the
3 times 10 to the 9th is 1 copy, and you need to multiply that times 2 for both mom and dad's contribution. And obviously, megabase, the next one is million kilobase, right? So that's 10 to the 3rd, and then you're just down to the bases after that. So 5 million bases is the minimum amount of material or content that needs to be affected for you to detect it. But that's not always guaranteed either, because if the banding pattern there is not very unique, like it's just a blank space, for example, so let's just say there's 10 megabases here, and you just so happen to delete a portion of it, you may not be able to tell that something is missing if the banding pattern is not informative. So this is what you need to remember. By karyotyping, that's the smallest size of DNA that you can detect in terms of a change. So the other things, translocations and inversions, is sort of natural, because here, if you have, for example, a chromosome, let me erase this. So if you know that this is the banding pattern for this chromosome, right? And then all of a sudden, this is normal. What you see in the patient is something that, you know it's this chromosome because of the banding pattern in the Q arm, for example. But what you see here is something odd, like you see that's like that. It's quite different, right? It's obvious here because I drew it. But imagine looking at 46 chromosomes and trying to say, oh, that's abnormal. And I recognize that this portion here is actually from chromosome 11. But this is chromosome, I don't know, 22 or something like that. Oh, there must be a rearrangement. So translocations, easily done. And then the other thing is maybe you know it is this way. But what you see in the patient is this band here, and then something like that. So it looks quite different, right? So perhaps there's an inversion, a pericentric inversion occurring right here in the chromosome. So you can easily detect inversions if they're large enough. So all of these things can be detected at the karyotyping level. But what fails in karyotyping is when you start getting down to very small kind of things. So CNVs, if it is 500,000 bases, which is quite large, right? In terms of what we deal with today, forget it. You can't detect it by karyotyping. But if it's 5 megabases and above, you might have a decent time of detecting it. But we know nowadays most diseases do not involve changes that are this big. There's probably a very small fraction that involve things this big. Trisomy 21, yes, that's something that's known. But for the most part, most diseases do not involve that big of a change. So that's why you need higher resolution techniques in order to detect that. So there is an alternative to karyotyping. Let's say we get down to 500,000 bases, maybe 100,000 bases. So there is an assay that works pretty well. And that assay is called FISH. So FISH stands for, any of the texts other than the FISH text, want to guess what this stands for? So fluorescence, right? in situ. Oh, so sorry, that's one word that I wrote. It should be in situ hybridization. I'm butchering the. Sorry, I'm butchering the spelling here in the writing. So fluorescence in situ hybridization. So let me just tag up here what karyotyping is, okay? It's 5 megabases. Maybe I should do a better job, right? Because this is on the video. Karyotype. Okay, so just keep that in mind. 5 megabases. So fluorescence in situ hybridization basically just tells you what the technique is. The detection method is by fluorescence. The process of doing the detection or the probing is in situ. It's actually on the tissue or on the stuff that you're working with directly. And it is based on hybridization. It's how the probing is done. Technically, karyotyping is an in situ assay, right? You are looking at the chromosomes as they are in metaphase. You are staining with giemsa stain in situ, and you're directly looking at the DNA as it is. That's in situ. Whereas PCR, things that we do by snapshot, is far removed from in situ, right? It's sort of looking at a fluorescent signal that is of a PCR product that is, that's 2 or 3 steps removed from that. So this, like karyotyping, is directly done on the tissue, the diseased tissue itself. And there's a number of ways to do fluorescence in situ hybridization. So you can either do it on, directly on the metaphase spread that I just explained about. So this will be Metaphase FISH. And you can alternatively do it also on the interface cells, which is what we do here. Why can't we do Metaphase FISH in our lab?
Fixed tissue.
We only work with fixed tissue. And I would shoot myself if we were to do Metaphase FISH, because that means we have to open up a cell culture lab, collect fresh specimen, do all of the business of karyotyping, which is very, very tough work to do. And with Interphase FISH, you can use dead cells, because what did I tell you? Most tissues that you acquire from the body are going to be in interphase, in their resting state. Even all cancers, that you only find a minor fraction of them undergoing active division and mitosis. So we deal mostly exclusively with Interphase FISH. The way it looks is going to be different. So on Metaphase FISH, when you do, let's say, instead of staining with a giemsa stain, which is a very generic stain, you'll stain everything purple or light pink or light purple or whatever. But in Metaphase FISH, you're only looking for a very specific signal on a very specific chromosome. So what it'll light up with is right there. 2 dots, because you have your sister chromatids there. You should look for 4 dots, right? Because there's the other contribution from the other parent as well. But here, you can actually look at this and say, oh, this looks like a pretty large chromosome. I think it's chromosome number 1. And it's actually on this portion right here, which is where I expect that gene to be, for example. And you can do it that way. But in Interphase FISH, when you look at the signals, what does it look like? How many copies would you see? 2 instead of 4, right? These cells haven't replicated their DNA yet, so they are only in the 2' state or the diploid state, and therefore you would have only 2 copies, 1 from mom, 1 from dad. Whereas in metaphase, you would have 2 sets of this, so that's why you see the 4 signals. So you just see 2 dots, but you have no idea what metaphase chromosome they're on because it's not in metaphase. So what you see is just basically complete kind of darkness, or maybe some autofluorescence green or so in the background, or brown, and you just see 2 dots like that. So how do we get to this state in terms of performing FISH? So let's go back and discuss about, first of all, how do you make the cells amenable for FISH? So for FISH, let's just talk about what we do in our lab, okay? So we have, again, a block. And you want to do typically 5 micron sections. Any guesses as to why we use 5 microns instead of, say, 20 or 40 or 50?
Because if it's thicker, the cells will overlap and it's difficult to see the signal, differentiate the signal.
Right. Does everybody understand that? So 5 microns is roughly the size of one cell / one nuclei. So you're going to get a nice sort of monolayer of cells. Yes, you're going to cut through some cells and section it through. But for the most part, you want to have sort of a monolayer of cells. Whereas if you did, say, 50 microns, you can see the difficulty in terms of viewing when you look down at the, in the microscope, is you see multiple layers of cells all overlapping on top of each other, and you won't be able to make sense of anything. So this would be like looking at a lymph node, for example, where it's just so crowded with cells that you can't get a nice monolayer out. So in this case right here, we do a 5 micron section, and we prefer this because we're not really going to mess too much with the architecture. But there's an alternative of doing Interphase FISH or FFPE FISH, where folks like at the Brigham, what they do is they do a very thick section, 50 microns or so. And what they do is they digest through, so I'm just going to draw scissors here. So they digest through, deparaffinize, and probably some elastase or some maybe trypsin or something that helps free up the cells and loosen them up, cut through the matrix, the stromal tissue, free up the individual cells, and then they do a cytospin and essentially deposit the cells anew down onto a glass slide to create a monolayer. So this is still going to be in situ because you're technically still probing, the signals as they are in the cells, like that. However, what you lose by using this technique is the very valuable information that we started out with, which is the tumor itself, right? So when we look at an H&E slide, like John or Valentina or I look at it, we know that, for example, there's tumor here. What happens in this case, if you muck up everything and kind of blend it together, is you lose this architecture right here. In order for you to do this method, you really have to rely on very high tumor cellular samples so that in the end, what you're looking at are mostly tumor, and maybe a very minor fraction of it is normal cells. But if you do it this way, you do a section through here, but you don't move the cells around, you keep them in place. So you can always go back to an H&E and say, oh yes, I need to focus on this area to make my diagnosis, because if I look at this area right here, that's going to give me a false positive result. The fish will work fine and well in this area, but you're not looking at the disease cells. So it goes back to that sampling issue that I talked about yesterday, about looking at non-junk things, the real things, in terms of making a diagnosis. So we don't like this method here. We do it on the 5 micron sections. So the first step that you need to do is to deparaffinize. And actually, I'll cut that back. You heat fix it first, right? So this is typically done overnight. I think we do 65 or something, right, in the oven. You bake it overnight. It helps kind of fix the tissue to the slide somewhat. And also, it goes ahead and, I guess, makes the paraffin a little bit softer and looser for the next step, which is to deparaffinize. And the deparaffinizing is done on our robot right now because we have high volume, but essentially it's a xylene step where it helps melt the wax off quite a bit easier. And the third step is pre-treatment. There are a number of things that are required in pre-treatment. First, it is heat. Second, there's an acidic component. And third, there's a pepsin reagent. And there's probably some salts and stuff in there, and some buffer, etc. So the heat, acid, and pepsin is needed because pepsin requires acid to kind of be activated in order for it to work. The heat, typically, in the old days when we did this, we just did a microwave and literally boiled the slides for about 5 or 10 minutes. But nowadays we're doing 80 degrees Celsius because the VP2000 probably you shouldn't be boiling on there. It'd be hard to get it to boil. So we just do 80 degrees and a little bit longer incubation, for example, to get the same effect. But the main point of this pre-treatment step is this, is that first so I'm going to draw the paraffin in, right? The first thing is to deparaffinize. So let's get rid of the paraffin. And this is still on the glass slide. But the next step is if I blow up one of these cells here, and here's the nucleus. You remember this is an interphase like this, but then it's very tightly wound. Certain things are loose, but there's still histones and nucleosomes or whatever here. So the point of using the pepsin and the acid and the heat is basically to denature these proteins. The histones and the proteins that are part of the nucleosomes and all of that architecture that is used to organize your DNA, your 6 billion or so nucleotides, is to organize it, but you want to now to deorganize it. You want to open it up so that now it's ready for probing. So that step essentially kind of gets rid of some of this and allows the DNA to relax even a bit more. And it gets rid of some of the treacherous, tortuous sort of environment in the nucleus so that, and also in the cytoplasm, so that your probes can make its way into the nucleus and therefore stain the targets that you're looking for. So this is a make or break step right here. So if you don't pre-treat well, you can either you probably will end up with no signals whatsoever because it doesn't give the probes a chance to do its thing once it's on the cell. So you pre-treat, you probe. Typically this is overnight. Depending on the protocol, 37, sometimes 40 degrees Celsius is what we use. And then 5 is next day you wash. 6 is you can mount and cover slip. I think these are all the steps here. So obviously any probe that's non-bound, you want to wash it away because that's the non-specific stuff. And then you just mount, cover slip it, seal it in terms of using a nail polish on the edges, and then you're ready to do the analysis. So we've left out one key ingredient is the probe. How do we generate the probe? Any questions so far about the technique? So the probe. For fish, any guesses what the resolution is? It's got to be better than 5 megabase, right? So hundreds of kilobase. So we've dropped down to probably more than an order of magnitude. Right? So an order would be around 500, right? 500,000. So we can go down below 500,000. So it's maybe an order and a half of magnitude in terms of increase in resolution. So to get this kind of thing is you have to be able to label a certain subsection of the genome that you're trying to look for, right? The probe has to be designed for a certain subsection. So let's say this is your genome. So I'm looking for just this region right here. And this region happens to be 300,000 bases. So you have to make a probe that's specific for this, which is on target, but not binding to the rest of the genome. So fortunately, from the Human Genome Project, we inherited a very useful substrate, which is bacterial artificial chromosomes. So during the Human Genome Project, which I'll talk a bit more later on, they developed these sort of very large plasmids, circular DNA that can be grown in bacteria. And the reason why they had to do that was they had to break down the genome into subsections so they can sequence it a little bit easier by Sanger sequencing. So it's sort of a hierarchical approach to deconstructing the genome so that they're in workable sizes. And because of that, now we inherit a certain clone that we know is this portion of the genome, because hopefully people have verified it by back-end sequencing. And we know it's going to target, for example, the EGFR gene that we're interested in. So what you do is you go online, you can order this back clone. It'll come as a circular DNA. It'll probably have an origin of replication, and are we using AMP for the resistance? Yeah, AMP, maybe sometimes some other resistance selection marker to grow this up very specifically in bacteria. So what you do is you take your E. coli. I think sometimes you have to transform it into the E. coli and then grow it overnight in LB broth. You plate it out, pick the colony, grow in LB broth overnight, 37 degrees. It's going to get confluent, and then you have this sort of pretty nasty tube of bacteria. Probably smells quite a bit, but that's what contains your plasmid or your bacterial artificial chromosome. So you probably have hundreds. These are probably not very high copy number. Hundreds of these, tens of hundreds in each E. coli. So you burst them open, and then you do DNA extraction like I mentioned yesterday. So remember the sonication or the mechanical or the SES. So it's the same process. Here is a little bit more challenging because these E. coli have cell wall. It's a bit tougher to get to them. So you probably have to use not only SDS but also sodium hydroxide is another portion of the buffer. But essentially in the end, you purify this out through DNA extraction, and you basically have this tube, highly, highly pure DNA, which only contains this portion of the genome, but nothing else. So this is where you get your specificity from, is that you're targeting just this region of the genome in this tube. So the next step is to make the probe. You use this as a template, and you do one of two things. One is called Nick translation, and the second is called random prime labeling. So I'll explain the first one. So imagine this is the double-stranded DNA, part of the large 300,000 bases in terms of the backbone. You're going to treat it with DNase 1 at a certain concentration, not very high, a certain sort of stoichiometry or concentration, so that it's going to go in and create nicks, single-stranded nicks in your template. Draw this a bit longer. And then you add into their DNA polymerase, which looks at these nicks right here. The 3' ends serve as essentially a primer. And what it's going to do is the polymerase has 5' to 3' exonuclease activity, and it's going to use the complementary strand as a template to produce a new daughter strand. So it's going to do this. So probably maybe one of these happens first. It creates a new daughter strand. And I guess I just drew them all overlapping, but it's going to do this almost randomly because the DNA's, yes, there's probably some bias, but for the most part, relatively random. So it'll do this, but what you want to do is you have your DNTP spike into here, but you're going to mix it with a proportion of DUTPs that's labeled with a certain color. So you could use FITC, for example, label DUTP, that will give you some green color. Or in a separate tube for another target, whatever, you can use some red labeling. And that's how you're going to label the DNA. So it does this almost randomly. And I'll keep the backbone right here for now. It does this almost randomly, giving you snippets, maybe a few hundred bases or so, of new nascent DNA strand that is now labeled. If you have an A on the complementary strand, now you have a U, depending on what the AT content is. And then these are going to be labeled with green. So essentially, the DNA is encoded, but then also containing your dye, which will be your probe that you use for the probing stuff. So random prime labeling is a little bit different. So what you're going to do is take your DNA template from the backbone, double-stranded, melt it off, and what you want to put into here are random primers, so they're random hexamers. So they're like 6 bases representing all combinations. So you'll have 4 to the 6 combinations of this. It could be ATCGCA, AATTCC, all combinations that will hopefully bind randomly to, not randomly, but complementary to where they are, but almost randomly so that it covers the entire backbone. And those serve as primers for polymerase, which again creates new daughter strand spiked with DUTP labeled green or red, and also give you some sort of a similar thing. You could do it in several ways. You could do initial lot with Nick translation and then just use random prime labeling of the initial probe to make more probe. You can do it that way as well. Or you can start from the original backbone material to do it. So either way works pretty well, but in the end you basically get a soup out. You have to clean it up, wash it, and pellet it and clean it up so that you get rid of all the non-specific dye and etc. But you end up with a tube of fluorescently labeled snippets of DNA that are complementary to your target, which is represented by the backbone. And at the end of the day, this is what you're doing, is that, let's just say we labeled it in green. So across this entire backbone, you're going to label stuff. So here's your ORI, here's your AMP. You're going to just label the entire backbone and insert. So it's going to represent everything covered, maybe 100 bases or so across the entire backbone. Yes, you're going to create something that's complementary to an origin of replication and also ampicillin. Hopefully the human genome doesn't have that, which it shouldn't. But the stuff that you're interested in will be the majority of the sequences, and that's going to represent your target DNA. And so we go back and basically summarize, which is fluorescence. This is where the fluorescence comes from. In situ, because remember we take the layer of tissue and we pre-treat it, we keep things the same, we open it up a little bit, we coax it to be a little bit more relaxed, but otherwise everything remains where they are, and that's why it's in situ. And then the next step is we put on the probe and allow hybridization to be the method of detecting your target. Once it's hybridized, it's all, or when it's hybridizing, it's all based on not random, but Watson-Crick base pairing, A to G, C to G. That's another level of specificity. So the backbone determines the specific target that you're doing, but the Watson-Crick base pairing based on hybridization is the additional step of specificity that's added to your system during the probing. Yes, you might get some off-target stuff, but the whole idea is this, is that you have your cell, your nucleus. Let's just say this is my target. You technically can't see it, right? I have one probe binding to it. So just one snippet of 100 base pair, maybe 5 U's are labeled. Can you see that by light microscopy?
5 U's?
No. 5 molecules of FITC, you probably wouldn't. There's a way probably that you can with a very high resolution microscope, but that's a very expensive system, hundreds of thousands of dollars. But yeah, 5 probes of used, you're not going to be able to see it. But if you have 1,000, so let's just say the target is actually this long here, and you just bombard it end to end on both strands, right? Both strands when you're melting it off with this. The collection of, say, thousands of probes, tens of thousands, 100,000, all together will shine pretty brightly. Yes, you're going to get some off-target binding in the nucleus, in the cell, and in the outside. Hopefully during washing, you're able to get rid of most of them. In the end, they don't amount to much. They give you a sheen of green, but overall, you won't see a collective signal that will be true. So when you look at it, you'll see a very true signal that's just spot on, very punctate signal, round, and that'll suggest that's where your target is. Make sense? So the things that you can detect by FISH. There are basically 3 things. 3 readouts. So if I have a cell here, here's the nucleus. I'm designing this assay like that. There are 3 things that you can get out of this. What's the first? Yep, so one thing, someone says gain or amplification, that's essentially enumeration, right? Counting. So you can get a number. I have 2 reds and I have 2 greens. Very basic. What's another basic thing? Location. The 2 greens are on top of the 2 reds. I can say that. And the 2 greens are separated by about 5 diameter probe distances. Similarly, the red also shows about 5 probe diameter distances. So location. And then something that I usually always say is not very obvious is the intensity, right? How bright the signal is. It's very hard to keep that consistent doing tissue section and things like that. So we don't rely on it as much. But there have been cases where we've seen like the red looks like this, and then the green looks like that. And across all cells, you see something like that. That's very unusual, and we've called that positive. Kind of blindly, and just recently with Zhongli's method, we showed that that is an out translocation. So it is informative when you get something like that as well, but we don't routinely rely on it. So you can get number, you can get location, you can get intensity. Number is easy, right? I just need to count how many greens and how many reds there are, and therefore I should be able to detect Polyploidy, Aneuploidy, and CNVs, down to hundreds of thousands of kilobases. A very good improvement, right, from karyotyping. Now we've gone down to an order of and a half of magnitude lower in terms of, or better in terms of resolution. Location, where things are. For copy number, we don't really care, right? We just need to know how many dots there are. But location can be useful when you're trying to detect translocations and inversions. So just very quickly, I have 2 chromosomes. Move this. Normally, this is gene, I'm just going to do BCR and then able. This is going to be hashed. So I have a true translocation, okay? I don't remember if it's Q or P or whatever, but just take my word for it that this is BCR able. So here, what you can do is maybe target the able gene and say, I'm going to design a probe, and I can order a back clone that represents the 5' end of able, and another back clone that represents the 3' end. It could be 2 flanking regions of the gene. So I'm going to label this in red, I'm going to label this in green. So when there is a break apart event due to translocation and interchromosomal exchange, switching partners here, then the green will go here, the red will remain where it is, right? Do you see now why location can give you some signature that something is happening with regards to rearrangement? So typically they will be together, so in a cell it will look like this. They'll be right next to each other, typically normally like that. Oftentimes they're on top of each other, and optically red and green makes yellow. So you'll maybe see a yellow signal instead of a red-green right next to each other. But in a positive cell showing translocation, you'll probably see one normal sort of germline copy, and then you'll see the other one completely apart, like that. So spatially, I can say that normally the signal should be together, but here I have a break apart event or a split event that tells me that there's a translocation involved here. You can design this assay the other way as well. So I could design this assay where I know this piece right here is involved in the rearrangement. This piece is also involved. So normally they are apart. But now when they come together, they're fused. So this would be a version of a fusion probe to detect a rearrangement. So in this case, this becomes this is normal, and this is abnormal. Okay? So when they come together, normal, abnormal. Sorry, normal, abnormal. So there's an advantage and disadvantage to either one. So if you use a break-apart probe, it's better because clinically it'll be more sensitive. If you use a fusion probe, you have to know the 2 partners that are involved. So it's very specific. If you cared about what is being involved here, then you would use a fusion probe. But in most cases, we don't really care. Like I don't care if ROS is fused to CD74 or SLC34A2. I just need to know that there's a breakpoint and a rearrangement being involved here. And therefore, all of our fish assays in the lab, we use break-apart probes because it's more clinically sensitive. At one point, maybe it's important to know what the partner is. So, for example, RAR alpha PML, that one will respond to retinoic acid. But there's another version, I'm blanking, I think it's the PLZ one, which does not respond to retinoic acid. So in that case, you do want to know the partner. So it's important maybe to confirm that finding with RT-PCR to confirm that, yes, it involves RAR alpha and PML. So there's just certain situations where you need that and other situations where you don't. So very quickly, for an inversion, it's a little bit different. Here's the chromosome. Here's the resulting one. The 2 genes are on the same chromosome. And let's say this one gene is like that, and the other gene is like this. So you see what's going on here? There's a switcheroo right here. The big gene is going to go like that, flip upside down. This is a paracentric inversion. So what you could do is design similarly. Let's say the big gene is where the culprit is in terms of the ALK receptor tyrosine kinase. So the green will remain here, but the red will now go up here. Here, right? So you have a splitting, but it's very subtle. On fish, you would look for something like this. So that's normal, 2 copies, diploid. The inversion was separated by about 2 probe links apart, maybe 3. But the thing is, this is an intramolecular event, right? So from cell to cell, you're going to see always something like that. So there you have positive cells around, and then you have, try to quickly do this. And these are the individual nuclei. You see how they're kind of consistently apart? That's how you can tell that there's an intramolecular or intrachromosomal rearrangement. Whereas if it is a true translocation with 2 chromosomes, you'll see them kind of variably apart. Sometimes far, sometimes close, sometimes midway in between, all kinds of flavors because there are 2 independent chromosomes or derivatives that are occurring. So I won't get too much into the details of how to score because I think that's that's more for like an interpretive sort of session. But there are certain thresholds that you would draw if you're doing copy number versus rearrangements to call something positive. There's sectioning artifacts, for example, so that's why you built that into the assay. But otherwise, this is a summary of FISH. So very quickly, to compare FISH to karyotype. Give me some advantages of FISH and some disadvantages. It doesn't have to be this many, but what's an advantage? I already highlighted it. Resolution, right? So better resolution.
Preserves the architecture.
Yep, architecture is preserved. More in situ, right, than karyotyping. What's another one? Is that? Yep, so you can use it on dead tissue. Basically meaning that you can do it on interphase. Anything else?
Turnaround time.
What's that?
Turnaround time.
Yeah. Turnaround time probably will be a lot faster. You can do this probably within a day and a half and get the results out. Karyotyping, you have to wait a few days or a week for the cells to grow, do the fixation and hypotonic solution and the dropping and the staining. You have to age it as well, actually, for a day or so. So it could take over a week to do the karyotyping. So what are some of the disadvantages?
You have to know your target exactly.
Right. So this one, it has to be, it's sort of a double-edged sword, right? It has to be very specific. You have to know what you're fishing for. It's not a genome-wide type assay, which is karyotype. Karyotype, you just roll your die and you just see what you get based on the 23 sets of chromosomes that you get out. But otherwise, you don't see anything else. But for FISH, it has to be very, very specific. You have to design from the get-go what targets you're looking at. So for the most part, it's very limited. You can only do 1 gene, 2 genes at a time, but really no more than that. But otherwise, you can get copy number, just like you would in karyotyping. You can do inversions. You can do translocations. You can do most of it. And in fact, there are certain things that you can do that probably is a little bit more specific in FISH if you were able to design it, if you knew the breakpoint. Whereas karyotyping, depending on what you see, what banding pattern, what day it is, it could be a hit or miss in terms of the results. Is that good for now? So we'll take a break.